Okay, next, right, you have looked at the signaling pathway, right, with the ligand receptor interaction, signal transduction, and lastly cellular response. So now we're going to look at how to fine-tune or regulate the strength of the cellular response, the type of cellular responses. Okay, so we remember that the signal transduction pathway, and really the whole signaling pathway is multi-stepped, right? And at each step, right, involving each protein, each molecule, right, each step can be regulated. So overall, there's going to be a very fine adjustment and really fine-tuning, right, of the strength of the signal and therefore the strength of the cellular responses in the target cell. Okay, so the first regulation of sorts is signal amplification, right? So in the whole signaling pathway, right, especially during the signal transduction, right, it offers many opportunities for the strength of the signals to be enhanced, to be amplified. How? That is through the activation of more than one downstream molecules from one upstream molecule. I hope you understand what's upstream and downstream. Because remember, right, the signal transduction pathway is in sequence, right? Say one protein activates the second one, second protein activates the third one. Right, so we have upstream, right, those before, and downstream, right, those that happens later, right, in the sequence. Okay, right. So in this picture, right, you see an example. We have one signaling molecule binding to one receptor. It happens to be a G protein coupled receptor or G protein linked receptor, and that will activate the receptor, which will activate the G protein, which functions as a relay protein here. Right, so activate a G protein, which will then activate the adenylate cyclase. Right, so one signaling molecule, one receptor, one G protein, one adenylate cyclase in this picture. Okay, but in this picture, right, the one adenylate cyclase produces 3 molecules of CMP. And in real life, wouldn't be just 3, right? In real life, as it's an enzyme, right? The adenylate cyclase is very effective, right? So it can produce quite a number of CMPs. But let's say 3, So one receptor, one signaling molecule, but 3 CMP, right? So in this case, the signal has been amplified, right? Has been this stronger, because there's now more downstream molecules, right, in the signal transduction pathway, right? 3 CMP. So not 1 to 1, now 1 to 3. Then with the 3 CMP, right, they bind to and activate 3 different protein kinase A. But each protein kinase A go on to activate 3 molecules of the second protein, right, in the signal transduction pathway. Let's say it's a second kinase. Right, so one protein kinase A activates 3 downstream protein. So 3 protein kinase A, right, obviously 9, right, downstream proteins activated, and all this from one signaling molecule. Right, so again, you see how the signal has been amplified again. Right. So cut the story short, right, subsequent steps, there can be more and more activation, or the activation of more and more downstream proteins, which eventually results in the activation of many molecules of a particular enzyme, which then can say, let's say, make many of a particular product, right, in the cytoplasm. Okay, so that is a very strong cytoplasmic cellular response triggered by one signaling molecule. Right, so whenever we have the activation of more than one downstream molecule or proteins by one upstream molecule or protein, right, then that increases the signal, the strength of the signal, and that is known as signal amplification. Okay, so this can happen with our second messenger, as you can see in this example. That can also happen during the signal transduction pathway, especially the phosphorylation cascade, as you can see here, assuming these are all kinases. Yeah, so one kinase can phosphorylate and activate more than one downstream kinase, which then phosphorylates and activates even more downstream kinases. Okay, so the signal amplification can be seen in a lot of different steps in the whole signaling pathway, but usually involving the second messenger and also involving the kinases in the phosphorylation cascade, usually. But then doesn't mean other steps cannot amplify signals, but usually second messenger and the phosphorylation cascade. So that's signal amplification, right? So making a much stronger signal and therefore much stronger cellular response from a small number of signaling molecules binding to the receptors. Okay, next we have the enhancement of the efficiency of signaling, because signaling is a multi-step, right, involving different proteins. So if you imagine, right, let's say the first protein here, MLK, is supposed to activate the second protein here, MKK7. Okay, so in the cytoplasm, right, they are floating around freely, right? So in order for MLK to activate MKK7, they must be by chance, or MLK must be able to by chance, right, bump into, or better term, effectively collide with MKK7, right? So that one is a bit by chance, right? So not so efficient, right? But we can increase the efficiency by something called the scaffold proteins, which are very large proteins that can bind to the different proteins involved in the same signal transduction pathway, right? So they can find each other more efficiently in the cytoplasm, right? So that will increase the efficiency of signaling and therefore increase the speed and usually also the strength of signaling. Okay, but then we can also trigger different cellular responses from just one type of ligand or one type of signaling molecule. Right, so one ligand will activate one receptor. However, right, during the signal transduction pathway, one of the relay protein can activate more than one types of a second downstream protein. Right, so previously, right, when we learned signal transduction pathway, we looked at one, say the first kinase activates just one type of downstream protein. So first kinase activates second kinase, then second kinase activates a third kinase. So this is one pathway. But what we are saying here is, right, the first kinase may be able to activate two different kinases. Right, so can activate, so let's say the first kinase can activate kinase 2 and kinase 3. Then kinase 2 can activate another kinase for another pathway. Kinase 3 can activate a totally different kinase for a different pathway. Right, so for every steps in the multi-step signal transduction pathway, if a particular protein or molecule can activate more than one types of downstream proteins or downstream molecule, then there can be branching, and that increases the number of pathways that can be activated from one signaling molecule, one ligand, and leading to different cellular responses at the end of the different pathways. So one signaling molecule can trigger more than one response in the cell. And here, right, with the so many different types of responses, different cells can have different responses depending on the type of signaling proteins and signaling molecules they have. All right. So first of all, the same ligand must be able to bind. They only bind to specific receptors, right? Right. So they only bind to receptors found on target cells. However, right, inside the cell, right, let's say these two are, if you follow my cursor, they have the same receptor. So the same ligand, right, can bind to both of these receptors because they're the same. But because the two different cells express different relay proteins, right, for the signal transduction pathways. So in the end, right, the same signaling molecule, same ligand, activating the same receptor, activates different relay molecules, and that leads to different responses in the two different cells. Then I jump to the last one, this one also, right? It's a different receptor, but it has the same ligand binding site, right, for the same signaling molecule. But because it's a different receptor, then like obviously activate a different relay protein and therefore different response also. Right, so this is how the same signaling molecule, same ligand, can activate or trigger different responses in different cells, right, depending on the types of proteins that the cell has, right, that can be activated by the same ligand or the same signaling molecule. Right, then lastly, I'm just going to talk about termination, because we have been talking about how to amplify, right, how the signaling leads to different responses, right, all very positive, but obviously we need to have a way to stop, right, to regulate, right, to lower the response if needed, right. So that's where the termination comes in. Right, so signaling molecules do not stay bound to the receptor like forever, right? They leave because the binding is not via strong bonds, right? Just like any protein binding, usually the binding is via weak hydrogen bonds, ionic bonds, for example. Right, so the signaling molecule, the ligand, do not stay bound on the receptor forever. Sometimes there can be other enzymes coming in to like digest the signaling molecule, break down the signaling molecule also. So once the signaling molecules is no longer bound to the receptor, right, the receptor will revert back to its original conformation, which is inactive. So then that will stop the triggering of any more signal transduction inside the cell. Then inside the cell, right, whatever that has been activated can be deactivated, right? So we mentioned second messenger. One example is CMP. The CMP is synthesized, right, in response to the binding of the ligand to the receptor, right? So the CMP also don't stay in the cytoplasm forever, right? They are removed by the enzyme phosphodiesterase, which converts it to AMP. Right, so once the CMP is removed, then whatever protein that CAMP binds to and activates will now no longer be activated. Then the signal transduction stops, right? And then we also mentioned signal transduction via phosphorylation, right? The phosphorylation cascade, right? So phosphorylation activates the protein, right? The kinases. Now dephosphorylation by the enzyme phosphatase would then inactivate, right, all these proteins or kinases. So again, stopping the signal transduction. Right, so once signal transduction stops, right, then obviously there's no longer the trigger for any more cellular responses. Right, so the cell stops responding, right, once the signaling molecule leaves and the rest of the activated molecules inside the cell becomes inactivated. So that's termination. Okay, the duration of the activity of the relay molecules affects the strength of the signal, right? So the termination don't happen like immediately, right? Obviously there is some lag time, right, between activation and inactivation or termination, right? So for example, right, with the ethanol cyclase, right? So ethanol cyclase is activated when the receptor is bound, right, by the ligand, and then there's in the example that we had earlier, right, in the previous video, then we have the G protein, right, which is activated, and G protein got activated ethanol cyclase. So the ethanol cyclase stays activated for a while. Right, so depending on how fast the ethanol cyclase is inactivated by the termination mechanism, right, then that determines how many CMP is made, and therefore how strong the signal transduction is, and how strong the cellular response is. Right, so the faster termination occurs, the faster the response ends, and also the weaker the response. So this is how we fine-tune, right, the response. We don't only enhance and amplify the signal, right? There can also be a way to lesser or lower the signal and eventually terminate the signal, right, which is this slide termination. Okay. So once the different proteins or molecules inside the cell, the receptor also, right, once they are inactivated, then this is like a reset, right? So the molecules, the receptor, the different proteins are ready for the next round of signaling when a new ligand binds to the receptor again, right? Then the whole triggering of signal transduction will happen again. But that can only happen again when the proteins and molecules involved have been inactivated first. So that also tells us the importance of termination. Right.